my computer.
Thank you, sir.
Okay, let us start. I guess uh, Janat may have some problem. Is the slide visible to you?
Yes, sir.
Yes, sir.
Very good. Okay, let me start. Today we shall discuss on genome editing, the concept, mechanisms, methods, and application. And genome editing is the latest uh, genetic engineering or bioengineering technology, uh, which has been used in almost all kinds of cells, from plants to even human cells. And in today's discussion, I shall discuss the basic concepts of genome editing and the science of genome editing, how it was evolved, and what are the various kinds of genome editing are being practiced. And also, I shall focus the CRISPR technology and more advanced base editing, prime editing, and also discuss some of the examples how we can utilize genome editing technologies for improvement of crops as well as other purposes. First, let us know what is gene editing or genome editing. Perhaps you know that what is genome. The entire genetic material of an organism is called genome. And the definition of gene, I hope you all know that gene is a segment of the DNA which can transcribe into mRNA and translate into protein. So genome editing, we can define as the techniques using site-directed mutagenesis to make small changes to DNA sequences at a known location. Uh, so it can be even in the RNA also. So a small change we can make by using the uh, smart technology, and we call them genome editing. Here I mentioned the site-directed mutagenesis. That means it is like a precise mutagenesis, that is making a mutation in the DNA. But genome editing is not only limited to the mutagenesis. The recent development allowed us to do even transgenesis and many more. But the widely used genome editing is the mutagenesis to make small changes in the DNA sequence very precisely to improve the trait of an organism. So genome editing, as I mentioned, uses site-directed nucleases to cut the DNA or to make any changes in the DNA. You need a molecular scissor. Molecular scissor to cut the nucleic acid, that is DNA, RNA, is called nucleases. That is the enzyme. to make a desired change at the specific location in the genome. That may either be a small deletion or a substitution or the addition of a number of nucleotides or bases. So here is the almost a complete definition of genome editing. That means genome editing can be defined uh, as the uh, editing of the genome using site-directed nucleases, and the editing uh, must be uh, very specific, and it can be a small deletion, a substitution, even maybe a small base deletion or nucleotide deletion. A substitution, sometimes substitution, if a wrong sequence is there, substitution can be happen, or the addition of a number of nucleotides or bases. Let me show you the illustration. This is a schematic presentation or illustration of the basic genome editing techniques. Here you can see the DNA binding domain, nuclease domain, and uh, we can use the DNA binding domain, nuclease domain, for example, Cas, a CRISPR-Cas9 or like that, and target the, for example, a plant DNA. I'm showing you the how plant DNA can be edited. So we make a construct using DNA binding domain like guide RNA and nuclease domain like CRISPR-Cas system. And then you allow this, construct to interact with the cell. So cell has the double-stranded DNA. So here, nuclease is just like the uh, molecular scissor. And DNA binding domain is a complementary guide RNA, which can find the specific target site of the DNA. So you put the address and put like postman locates the address and then drop the letter like nuclease card the double-stranded DNA. So after cutting the double-stranded DNA, uh, three things can be happen. One is SDN1, site-directed nuclease 1. Another is SDN2, and third one in SDN3. That means three types of editing can be possible. The first type is just you cut the double-stranded DNA in a specific location that is on the desired gene, and then you do nothing. That is, you allow to rejoin because DNA has an inherent mechanism of repairing. So if by any means DNA has an injury, here double-stranded break, the two parts immediately tends to join together. During joining, here you can see this red nucleotide means this nucleotide, for example, uh, here deleted or changes. That means one nucleotide, if changed in the sequence of a gene, if it is a lethal gene or, for example, disease susceptible gene, then these minute changes can result in uh, affect the expression of the gene. So may inactivate the function of the gene. As a result, disease resistance can be developed. So this is very simple. And here you can see no transgene is introduced. As a result, it is like a mutation breeding we do conventionally by gamma radiation, or EMS. Uh, but here is the precise, and nothing is added. This is why like natural mutation taken place. And SDN1 in many countries are allowed to introduce in the farmers field directly without biosafety guideline. The second one is SDN2. Here, after the double strand break, you put a template of sequence. A template means you desire here um, this side and this side, the green, matching with the DNA sequence in two parts. And in the middle, you make some changes in the nucleotide sequence. And usually we do this type of changes considering the, some bad mutation uh, occurs compared to the normal, organism. So you just make a correction. So then it joins here and ultimately change the DNA sequence. Here, three nucleotides uh, is corrected or edited. The third one, and here also no nothing is new gene from the other species introduced here. Only you made some minute correction without adding any foreign gene. And it is also non-transgenic. So SDN1 and SDN2 are non-transgenic. SDN3 is different. After the double strand break, you can put even a new gene to add here, like we do the transgenic plant. So it can be transgenic. And nowadays, this SDN3 requires, if it contains a new gene, if you want to introduce and make the transgenic, then you must go through the biosafety guideline. In Bangladesh, our government introduced a standard operating procedure for SDN1 and SDN2 so that the gene edited plants can go directly to the farmer's field without rigorous biosafety guideline. There are some committees nationally, they will just check your data. But in case of SDN3, it is a bit complicated and not well accepted uh, because of transgenesis or like that. Apart from SDN1, SDN2, and SDN3, genome editing is expanding at the methodology level, now even at chromosome level, or very, robust, editing can be possible. That is, here you have seen only one place or on one gene, it is changes. It can be multiplexed. That means at a time you can edit DNA in several locations. Multiplexing is possible. And even we can edit a single base, or even we can do prime editing. I shall discuss later on. So major genome editing technologies are zinc finger nuclease, which was developed in 2005. This is one method. A second one is transcription activator like effector nuclease, which was developed in 2009. And the latest one, CRISPR-Cas system or CRISPR-Cas genome editing technology developed in 2012 by two very famous scientists, Emmanuel Charpentier and Jennifer Doudna. And they received the Nobel Prize in chemistry in 2020. Uh, Really amazing technology. And after 2012, This technology became a crazy topic for research worldwide, and huge progress been you know uh, observed. For example, nowadays in USA, CRISPR-Cas genome editing is used to treat the sickle cell anemia in human, like gene therapy in vivo. The remarkable scientific article, a programmable dual RNA guided DNA endonuclease in adaptive bacterial immunity. This is the Nobel winning research article published in Weekly Science in 2012. Here you can see adaptive bacterial immunity. That means CRISPR-Cas system. Basically, CRISPR means clustered, regularly interspaced, short palindromic repeats. Uh, CRISPR and CRISPR-associated protein, that is Cas, they are basically uh, discovered in bacteria and archaea as the adaptive immune system against virus and plasmids, because bacteria and virus, they also uh, attack by the viruses, bacteria and the archaea. So these prokaryotes, over the evolutionary process to fight back against the virus and plasmid. They cut the DNA from the virus genome and then put into the own genome those who survived during the attack, and uh, evolutionary developed the molecular scissors so that next time similar virus, if attacks, then they can cut the viral DNA or RNA. And it is over the years, it has been proved that a fantastic adaptive immune system of bacteria and archaea. It is important to mention here that this CRISPR-like strange sequence was first observed by a Japanese scientist, Ishino, when sequenced a bacterium DNA uh, and published in Journal of Bacteriology in, uh, you know, 1987. So from 1987 till 2011, scientists uh, only rotate on the story of adaptive immune system, and they precisely confirm this molecular mechanism. But Emmanuel and Jennifer, they met in a meeting in Puerto Rico in 2011, and during discussion, they decided to collaborate and try to see whether same molecular scissor, a natural molecular scissor, can be utilized to do gene editing in the laboratory condition in the test tube. And they were successful, and this is the article. So, what is CRISPR? CRISPR clustered regularly interspaced short palindromic repeats. Palindromic means, for example, Papa, if or how to say, when you reverse it, not Papa, for example, uh, any the sequence, if you reverse it, it remains the same. So CRISPR really amazing, and the CRISPR present in the bacterial and archaeal, many of the bacterial and archaeal genomes scatteredly. Uh, and CRISPR-based genome editing technique uh, that targets a specific section of DNA and makes a precise cut or break at the target site, and can do at least one of two things: make a gene non-functional, um, or replace one version of a gene with another. If there is an error, you can correct it. This is very simple output of genome editing, but it can be much more, and nowadays it is becoming user friendly. So I mentioned CRISPR-Cas technology is superior and smart, the latest one. And why it is superior than the zinc finger nuclease and the TALEN? The original implementation of zinc finger nuclease and TALEN site-directed nucleases for genome editing has proven to be cumbersome. Why? Because in both cases, it requires sophisticated protein digestion, synthesis, and validation. And if you want to learn more, JFN and Talent, you can find lots of literature in the different websites, even in the Wikipedia. You can see how complicated. But several organisms or cell lines have been successfully edited by ZFN and talents, but not user friendly. On the other hand, CRISPR-Cas9, uh, Cas9, there are many enzymes. Cas9 is very popular. Uh, and CRISPR-Cas9 or other CRISPR-Cas system are cheap, relies on single guide RNA. Single guide RNA means you synthesize the guide RNA depending on the sequence of the gene you are targeting to target the site specificity. Cas9 is a protein you cannot, synthesize. It is naturally originated, and it is a naturally derived technology. So this is another beauty of this technology. Can be readily designed based on genome of choices and in multiplex fashion in which several genomic loci can be perturbed or edited at the same time. This is really fantastic. You can, target several genes to correct or edit at a time, and it is called multiplex fashion. What you need to do, depending on the gene or the genomic loci you are targeting, you design the guide RNA and make the CRISPR Cas9 complex. So at a time, many places you can edit. System create stable and heritable mutation. This is very interesting. When a CRISPR-Cas system used to edit the DNA of an organism, it is stable and heritable, not like, the hybrid. So if in case of plant, if you edit a plant and offer the seed to the farmers, they can multiply year after years as it is heritable and stable. Transgene-free plants in few generations, because first generation CRISPR gene can remain, but within few generations, two or three generations, you can get transgene-free plants. That is the genome edited plant. Edited plant or variety can be used for plant breeding. For example, you edited a plant, but the editing was successful, uh, but the plant is not, edited plant is not commercially, profitable. In that case, you can use that edited line as a material for plant breeding to cross and back cross and transfer the gene to the target plant. Commercial plant. New set of CRISPR-Cas systems such as Cas12 or Cas13A, lots of CPF1, they are being discovered from many bacteria. So what is unique in CRISPR-Cas technique? Derived from a natural process I mentioned before, found in bacteria to protect themselves from the pathogen, like virus or plasmids, mobile DNA. Targets genes for editing and regulating, uh, comparable to Photoshop. Here you can see the system. For example, you design this part of guide RNA, Cas9 commercially available. So guide RNA you design, and then a CRISPR system, together, they work like, find the your target sequence based on the synthetic guide RNA, and then make a double strand, unzip or make the double strand break. Really a simple technique. And here, a simple illustration. Here you can see yellow guide RNA you targeted, and matching genomic sequence of the guide RNA of what you are targeting. And this is red one, the Cas9. So CRISPR-Cas9 system, the construct you developed, and then cut the double strand like this way. And after cutting the double strand, two things, gene can be disrupted, or gene has a new sequence. You can put the template, and sequence can be changed. So, and lots of other things can be done. But this is the simplified uh, principle of CRISPR-Cas system. I shall stop after explaining this one. Here is the various kinds of genome editing and manipulations by CRISPR-Cas system. Here you can see guide RNA, Cas9 system, and the full complex CRISPR-Cas9, or even you can change the enzyme like this way. And here, the double strand break is the ultimate target in a specific location where you are targeting. And there are two pathways involved in genome editing using the CRISPR-Cas system. One is non-homologous end joining repair pathway, and other one is homology-directed recombination pathway. Recombination means something combined to the original DNA sequence. And here, during repair, some mutations or deletions, or even correction of the genes, insertion or replacement can be taken place. Here you can see just cut and indel mutation, that is one nucleotide mutation taken place, which is shown in red. And here you can see two guide RNA were designed and cut into two places, and whole gene, from the two part cut, and it is out, and then, you know, uh, repaired, and gene deletion occurred. And here a template is put and cut into two places, and in the gap you put the correct gene sequence. So in these three cases, no transgene is required. We can call mutagenesis. So mutagenesis by precision system of CRISPR-Cas system. And they are in many countries, the resulted, the organism, especially the plant or fish, are allowed or receives the green pass to release to the farmers. In USA, in Japan, some plants and even fishes are being released through this process. For example, soybean, camelina, waxy corn, or white button mushroom are released in USA. In Japan, gamma amino butyric acid enriched tomato, and one fish, red sea bream with higher muscle content, and tiger pufferfish with higher muscle content being released in Japanese market. On the other hand, HDR pathway, here is simply, transgenesis, gene correction or gene insertion or replacement can be possible. So HDR a bit complicated, and due to the uh, restriction of the practical application, this process is slowly progressing. But the non-homologous end joining repair pathway is becoming a more popular and user-friendly technique for improvement of the uh, plants and other organisms. For example, in case of sickle cell disease, uh, the non-homologous end joining a single indel mutation is used to treat the sickle cell genetic disease. So here I would like to show you how to initiate. Here you see single guide RNA you design based on your target gene sequence, and then Cas9 nuclease, that is enzyme you combine, and ultimately Cas9 a complex is developed with the guide RNA. Here you can see you have given the address, that is guide RNA, where to cut by the Cas9. Here is the target, for example, target sequence. And here you can see it comes here and gene of interest, and then make a double strand break. Here you can see double strand break. And finally, when the naturally repair, uh, the repairing mechanism also cut some more nucleotide. And finally, here you can see the edited one has quite different, mutation in the target size. So this is all about, the principle of CRISPR-Cas system. And uh, now I can take a pause to get some of your question. So you may ask question now. Please ask question freely.
Sir, thank you for your nice class, sir. Sir, I want to know SDN2, sir. Again, I missed
Mhm.
it. Yes, sir.
Yes, you want to know SDN2. Let me go back. SDN2, what is your question? Yes, SDN2.
Yes, sir.
Is it visible now?
Yes, sir. Visible.
Okay. SDN2, after the double strand break, DSB, by the CRISPR-Cas system, uh, depending on your guide RNA in the specific location, you can put uh, a template. Template is if a gene is somehow mutated and became incorrect, so you can correct it by like this way, one or three, two nucleotide maybe wrong, uh, then the normal uh, natural organisms due to some mutation. Uh, so what you can do, you can put this type of segment. Then during repair, this segment can fill up the gap, and you can see the corrected sequence you can obtain. And here, no transgene is added, but you corrected for example, a spelling mistake in a word. So you can correct it. Or even a word is, wrong word in a sentence. So you can replace the correct one. Uh, and how do we understand when we compare the sequence with the, reference genome? Then you can find this error. After diagnosing the error, then you can correct it. Is it clear, Raihan?
Yes, sir. Thank you, sir.
Okay, great. And Jannat?
স্যার যখন আমরা একটা টেমপ্লেট দিয়ে এটাকে রিপ্লেস করছি তখন কি এটা স্যার ন্যাচারাল থাকবে না জিওমো হয়ে যাবে স্যার?
It would not be a GMO. It is not treated as GMO because In the uh, naturally, the gene was somehow disrupted. So you are taking it back to the natural. For example, in some of our gene, if somehow due to radiation or any exposure to the carcinogen, deleterious mutation taken place, then you can correct it, uh, and taking it back to the natural one. This is why it is not called the GMO, because you are not taking something from any gene from other species, just to make the correction and back to the normal one, standard one for the expression of the the gene. If somehow some gene is defected by any means, then you can correct it. Is it clear?
Yes, sir.
Okay. And how
Sir,
about, yes,
sir, sir, in case of SDN2, when we find that there is problem, and we correct it by correct template. So
yes.
from where we can collect the correct template?
Good question. What you do, we first by sequencing, you first confirm the error. Uh, what is the error? Error means somehow artificially changed from the natural sequence of a gene, and then you correct it by putting the right sequence. Here you can see uh, here are green and here are green, only the middle part was wrong. So this nucleotide fragment you can synthesize in the laboratory, so you can even order a company they will send you like the primer. Okay, and if you put it during the uh, editing, then naturally it fills the gap and synthesize the you know uh, a complementary sequence, and ultimately a ligase enzyme can paste it and complete it. Clear?
Yes, sir. Clear, sir. I got it, sir.
Yeah,
How about Sujan?
yeah. And thanks, sir, for your sharing. Sir, actually, genome editing facility is available. Service is available in Bangladesh.
Yeah, in Bangladesh,
This
genome
CRISPR.
editing facilities in several labs are available because this technology is very cheap. Like the PCR, you can do it in any laboratories with less equipment. The biggest challenge is the, in case of plants, transformation technology. If you have a transformation technology, then that means you need to have a tissue culture technology with transformation vector like agrobacterium, tumefaciens, or gene gun. We have the gene gun. Then you can do it easily. And in case of fungi or bacteria, if you want to do genome editing, no need tissue culture technology, you can very easily do in any laboratory. Um, in our country, NIB, BRI, and also Dhaka University, they did developed many edited lines. In our case, our students visited UK, and in Sufyan's and Nick labs, they edited the plants of wheat and screened lots of seedling 7,000 edited lines, because wheat is very complicated. Each gene has 6 copies, and they screened all the 7,000 and brought some of the edited lines in our lab. We checked and we screened, and we also did some of the work. And gene editing, if you want to do in other organisms. For example, in case of human or animal or fish, you need to use at the germline level. After fertilization, you can edit because plant has totipotency. On the other hand, adult cell of animal you cannot edit it. So at the germline level, if you edit them, then in vitro,
Right.
uh, how to say, in vitro fertilization and then edit. And after that, you can put into the, in case of fish, I guess, after fertilization, you can get, after editing, you can get the uh, fish fry or like that. And you can get the edited, uh, uh, you know, a fish. And in case of other animals, for example, cattle or sheep or goat, like human, uh, in China, uh, one scientist, he edited at the germline level after fertilization, the embryo edited, and two babies, Nana and Lulu, was born. He edited for the HIV resistance because both parents carries the human immunodeficiency virus. So they were scared about their future baby. So they got two babies with editing of the susceptible site of the DNA, and the babies are resistant to HIV. But as human editing at the current stage for at the germline level is strictly prohibited, he was sent to the jail, and at that due to the in vivo human editing, there are some delicacy off target. If there is any off target, it can make severe consequences. But anyway, in future, maybe when the accuracy would be 100%, it could be applied. In case of human, only sickle cell disease anemia, that is using SDN1 and single nucleotide error, has been corrected in US since last year. So they gave the permission. But in other countries, there is only one case in UK, blood cancer patient Alisa. Uh, one base was defective, so it was base editing resulted Alisa free from the blood cancer because she was at the fourth stage of her cancer. There was no hope. But even though after the success, gene editing uh, has not received the uh, legal permission to use in human worldwide. But because of some, still there are some off-target and some, methodical delicacy. But in case of plants, you can find plants, microorganisms, even fish, thousands of literature you can find. But in case of a human in vivo, really delicate, ethical, and other serious concerns restricts to give the permission. But in future, maybe the situation will be different as the methods are moving very fast. Any other question?
Thank you, sir.
So if you have no question, then I would like to stop here. That is various kinds of genome editing and manipulations by CRISPR-Cas system. And the next class we shall discuss more. But for your exam, this basic principle, please try to go through very carefully. I tried to make this illustration simpler. And in fact, this illustration was prepared by the peon MLSS Ashraful of our Faculty of Agriculture. He is very powerful in uh, using the Illustrator and Photoshop. So my hand drawing, he converted like this way. So this is, I think, for anyone, simple to understand clearly. So if you have no question, do you have any more question? Okay, if you have no question, then I shall stop here. And thank you so much for attending. And hopefully, next class we shall have in-person class at BSMRU. And I shall record this one and share with you. Thank you. Good night.
Assalamu alaikum, sir.
Wa alaikum salam.